The movement of water in xylem is caused by transpiration pull. Water is a polar molecule, so transpiration pull is facilitated by cohesion and adhesion. Cohesion is the attraction between water molecules, and adhesion is the attraction between water and the wall of xylem. These attractions are caused by hydrogen bondings. Monosaccharide, triglyceride, and fatty acid only contain carbon, hydrogen, and oxygen. Nucleic acid contains carbon, hydrogen, oxygen, nitrogen, and phosphorus. Phospholipid contains carbon, hydrogen, oxygen, and phosphorus. Amino acid and protein contain carbon, hydrogen, oxygen, nitrogen, and sulfur. DNA is replicated by semiconservative. Each parental strand is a template to produce a daughter strand by complementary base pairing. The hydrolysis of a triglyceride means to break down the fat molecule into 3 fatty acids and glycerol. Water is produced in condensation. Cellulose is made of beta-glucose, while starch is made of alpha-glucose. Cellulose is an unbranched, straight molecule, while starch can be branched and helical. Amylose is helical, and amylopectin is branched. The diagram shows the structure of insulin has 2 polypeptides. These 2 polypeptides are linked together by disulfide bonds.
The diagram represents the metabolic pathway that converts threonine to isoleucine. Once isoleucine is formed, isoleucine inhibits the enzyme threonine deaminase. This is called end product inhibition, and it is a type of non-competitive inhibitor. Isoleucine binds to the allosteric site of the enzyme, and it will change the shape of the active site of the enzyme. Enzymes are biological catalysts to speed up reaction. Enzymes have specific active sites, which bind to specific substrates. Some enzymes work inside cells, and some enzymes are secreted from cells and work outside cells. In polymerase chain reaction, high temperatures are used. 95 degrees Celsius is used to unwind the double helix of DNA templates. 60 degrees Celsius allows primers to bind to DNA templates. 70 degrees Celsius is the temperature for the Taq polymerase to replicate DNA. Taq polymerase is a suitable enzyme because they would not be denatured at such high temperatures. Non-coding DNA refers to the DNA that does not code for proteins. Alleles, genes, exons are all coding for proteins. Promoters are the DNA sequences at the beginning of genes, while telomeres are used to protect chromosome structures.
Table shows that 3 amino acids are coded by 4 different codons. When there is substitution mutation on a DNA strand, the codon may change to another codon. Out of these options, only option D will lead to the same polypeptide being formed at translation.
Phloem sieve tube cells are living cells. However, the cell does not have cellular contents, for example, no chloroplasts and no nucleus. But it still has cytoplasm. Lignified walls are specific structures in xylem. Mr. Miller and Mr. Urey tried to simulate the conditions of the early Earth to study the origin of life. In this experiment, several inorganic compounds were added into the experimental setup, for example methane, ammonia, hydrogen, and water. These inorganic molecules reacted in extreme conditions, such as extreme high temperatures. These inorganic molecules turned into organic molecules, such as carbohydrates, proteins, and nucleic acids. The image shows a group of enveloped viral particles. Structure X is the viral envelope. The envelope is derived from the membrane of the host cell when a new virus was budding out from the host cell.
Ions move across the membrane of a neuron. This is the movement of sodium ions. The 3 sodium ions are transported out of the cell by active transport because ADP is involved. The sodium ions then move back into the cell through channel protein. This movement is facilitated diffusion.
Embryonic stem cells is a type of pluripotent stem cells. They can form any cell type except placenta and umbilical cord. Multipotent stem cells can only form a group of related cells, for example, bone marrow can form a group of different blood cells. Totipotent stem cells can form any cell types including placenta and umbilical cord. Under normal circumstances, specialized cells can't differentiate to form another specialized cell.
Glycoprotein is a transmembrane protein that embeds in the phospholipid bilayer. This part of the glycoprotein contains the carbohydrate chain, which is essential to the function of the protein such as cell marker, cell adhesion and cell recognition. Tyrosine kinase receptors are found on the membrane of liver cells or muscle cells. This receptor is specifically for insulin to increase the glucose permeability in liver cells and muscle cells. The graph shows the effect of methylation on the expression of MT1E, a gene involved in the control of prostate cancer development. DNA methylation is a way to regulate gene expression by inhibiting DNA transcription. This is shown in the graph that the one with methylation has a lower expression than the one with unmethylated. This will increase the chance to develop prostate cancer.
Histones are proteins that are found in eukaryotic cells. Histones allows DNA to condense to form chromosomes.
Phototropism refers to plants growing towards light sources. It is controlled by auxin, which is produced at the tip of plant shoots. Auxin will be transported away from light to the shaded region of the shoot to promote cell elongation.
Fruit ripening is caused by a plant hormone called ethylene. When a fruit ripens, more ethylene will be released, so more fruits will ripen. This is the positive feedback. Karyogram shows a chromosome abnormality in a human female. There are 3 copies of chromosome 21 in the karyogram. The reason for having an extra copy of a chromosome is due to non-disjunction. Chromosomes stick together and cannot be separated in meiosis when gametes are formed.
A dichotomous key was used to classify the animal in the diagram as an arthropod. The antennae are pointing sideways. There is 1 pair of legs in each body segment. Basis sequences are used to determine the evolutionary relationship of 5 animal species. So we don't see any morphological differences between these species. There are 4 clades in this cladogram. Although species X and Y evolved from a common ancestor, we cannot show how many base sequence or amino acid sequence differences between these 2 species. The water vapor from the evaporation of water from mesophyll cells will diffuse out of the leaf through stomata. This process is transpiration. Transpiration will lower the water potential in mesophyll cells in leaves. So water will be transported up to the leaf through xylem. This is called transpiration pull. This is the direction of blood flow. So this blood vessel is an artery, and this is a vein. Artery carries high blood pressure, so due to the high blood pressure, materials move out of the blood to form tissue fluid.
Peristalsis is an involuntary action to push food moving along our intestine. It is controlled by the enteric nervous system, which is involved the spinal cord only. Innate immune system refers to a quick, nonspecific immune response. T cells and B cells are lymphocytes which are involved in specific immune responses. They will help the body to form specific antibodies. Phagocytes is a component of the innate immune system because phagocytes engulf any antigen by phagocytosis.
When there is a rise in body temperature, we have to rely on negative feedback to cool down the body to maintain a constant body temperature. There are several ways to cool down the body, for example, sweating. Vasodilation is another way to cool down the body. This will dilate blood vessels to allow more blood to reach the skin's surface to increase heat loss.
Female grasshoppers have XX sex chromosomes, and males have one X sex chromosome only. A recessive mutation appears in the X chromosome of a male. When this affected male crosses with a normal female, a carrier female is produced in F1 generation. When this carrier female crosses with a normal male, an affected male is produced in F2 generation. The graph shows that the number of hummingbird species in South America has increased continuously. Formation of new species is caused by speciation when there is selective pressure. Selective pressure includes competing limited resources, changing environmental conditions, and other limiting factors for the growth of population. Polyploidy is a condition where an organism has more than two sets of chromosomes. A polyploid plant reproduces asexually, produces male and female gametes, and crosses with a diploid plant to fertile offspring, are all facts for polyploidy individuals. However, they are not evidence for the cause of speciation. Fertilization can occur between polyploid individuals because polyploid individuals are reproductively isolated from the original diploid population, which means they cannot produce fertile offspring when crossed with diploids. This reproductive isolation is a key criterion for speciation. So the ability of polyploid plants to interbreed with each other but not with diploids provides strong evidence that a new species has formed.
Yersinia pestis can produce ADP in the presence or absence of oxygen. This organism belongs to facultative anaerobes. Obligate anaerobes can only produce ADP in the absence of oxygen, while obligate aerobe can only produce ADP in the presence of oxygen. Ingestion of food is an exclusive feature to holozoic nutrition. Fungi and bacteria mainly use saprotrophic nutrition to decompose dead organic matter. Holozoic and saprotrophic nutrition need to obtain organic matter from other sources, so they need to use enzymes to break down the organic matter. So this is the only common feature in these two nutritions. These temperatures are the preferred temperature ranges for these three different trouts. These three ranges overlap, showing their niches also overlap. Although they overlap, each trout can also live in some specific areas. For example, lake trout can live in colder water, so they can avoid competition with other trout. However, just based on temperatures cannot tell the condition of the niche for each trout.
Density-independent limiting factors can affect the growth of a population regardless of the population density. Forest fire is one of them because it will happen no matter the population size is large or small. Predation is density-dependent factor for prey population because it would be easier for predator to prey when the prey density is high. Climate change and eutrophication are anthropogenic. When the human population increases, more greenhouse gases and chemical will be released. Therefore, other populations will be affected by these changes. So they are also density-dependent factor. Allelopathy is a strategy in the relationship of competition. It means the organisms produce secondary metabolite that are not essential for basic functions, but to influence the growth of other organisms, such as inhibit and egg cell respiration. There are 5 assumptions for Hardy-Weinberg equation. The population size is large. The population is isolated, mating is random, no natural selection, and no mutation. Domestication of dogs means the process of changing the dog's traits for human uses. So involves selective breeding. When wolves have some favorable traits that are useful for humans, they are selected to breed to pass on the traits to next generations.